Welcome to today's Fungisphere talk here at the University of Sydney. I'm Dee Carter, Professor of Microbiology in the School of Life and Environmental Sciences here on Gadigal land. Today we're very honoured to have Dr. Ben Schultz, who is Professor of Biochemistry at the University of Queensland. Ben has a passion for yeasts and for beer, and today he's going to combine this passion telling us all about the metabolic diversity of Australian native wild yeasts and their use in making interesting new beers. So, Ben, take it away for us.
Thank you very much, Andy, for the kind introduction and for the invitation to speak into this Fungisphere series. Sounds really, really fun. What I'm going to present today is some work that we've been doing over the last couple of years, really a snippet of that work on understanding a little bit more about the metabolic diversity and utility of Australian wild yeast, which is a very broad topic, but specifically what we've been doing in that space about capturing, characterizing and using wild yeast for beer brewing. So this is work that is now out in a preprint on the bioRxiv with that same title, Capturing and Characterizing Wild Yeast for Beer Brewing. So you can go and have a look at the details of that if you wish. And this is work that is basically a core part of the PhD project of Ed Kerr, who was in my lab for his PhD and then a bit of a postdoc, and now has recently moved on to bigger and better things at CSIRO. And it was also in collaboration with Mark Howes, who is a doctoral graduate, is a PhD graduate from the University of Queensland in cell biology, but then a few years ago, a decade or so ago now, realized that his passion was in brewing, and so he founded Newstead Brewing, a nice craft brewery in Brisbane. And then he was keen to collaborate with academic research, and so got in touch with us several years ago, and was then part of Ed's PhD project in finding out what wild yeast could do to help interesting craft beer brewing. So before we get into the mycology of things, let's take a step back and introduce and overview the beer making process, because there are aspects of this process that are kind of important for thinking about the metabolic demands of the yeast in the fermentation stage of this process. So beer making is an industrially really ancient and really established process, and basically uses grains. So that's mainly barley, but also with some flexibility there. Some types of beer can have a bit of other grains as well, so wheat, for instance, or also other adjuncts, anything that can provide some eventual fermentation stage of yeast. So that might be things like corn or cane sugar, or rice, depending on where you are in the world. But the main core ingredient is barley. And then also later on in the process, there's water and hops and yeast. They're the key elements that come together to eventually give beer. But there's a whole lot of processing that happens to those agricultural, biological inputs to get from those raw agricultural inputs to the final beer product. And the first of those is the harvesting of the barley seed, and then the so-called malting of that barley seed. So this process basically replicates the initial germination stages of the barley seed. So barley seed, like all plant seeds, it contains the seed, the embryo of the new plants. And also in those seeds are stores of nutrients and energy that are required for the growing, developing embryo. So in the context here of beer making, the key nutrient stores there are starch as a store of glucose, and also proteins as a store of amino acids. And so both those glucose and amino acid stores are present in the seed. That's why they're really nice, valuable food goods for animals like us to eat. But for the point of view of the barley plants, the reason that those seeds contain those nutrients is to allow the growing embryo access to those nutrients during its early stages of growth before it can reach the surface and begin to photosynthesize for itself to make its own energy and and harvest its own nutrients. And so the process of malting then takes those seeds, makes them a little bit wet, and gives them time in the dark to begin that initial germination stages. So the seed soaks up that water, and then that stimulates a variety of different processes, transcription, translational processes, as well as activation of already existing enzymes in the seed that begin to break down those stored nutrients. So starch is degraded by amylases into maltose and oligosaccharides. The proteins and the seed storage proteins begin to be digested by proteases into small peptides and amino acids, and there are also other processes that go on there as well that are associated with opening up of the seed husk and making it more softer and more accessible from the outside, and also amenable for the developing embryo to be able to grow and breach out of it. So normally, if that seed were to be planted or to fall on the ground, then that process would continue, but the growing embryo would use all of the glucose and amino acids, put them into using energy and rebuilding new proteins for the developing embryo, and then it would begin to grow. But instead, in the process of malting, that process is only really just started, and then just as the seed begins to sprout, the process is stopped. Because what we want here is not for the developing embryo to use all of those nutrients, glucose and amino acids, but instead just to make them accessible and ready to be able to use for the yeast during fermentation. So this malting is a partial germination, but then it's stopped only after a couple of days. And then the growing shoots are sheared off. The seed or the malt then is dried to stop any more enzymatic activity. And typically then this is also kilned, so heated to roast it, to add some additional flavor elements if that's a desired part of the final beer product. And then it's ground to a relatively coarse flour or grist. And so that then produces the malt, which is perfect now for a hot water extraction in what's called the mash. So here, water is added, and so is the ground malt, and it's basically a hot water extraction where the starch and proteins that were in the malt are now solubilized into the hot water, and the enzymes which were translated and transcribed and activated during this malting step are also solubilized and able to access the substrate. So for instance, we have amylases that are able to access and digest the starch, and digest that into oligosaccharides down to maltose and a little bit of glucose. And the proteases that are here are able to continue the digestion of the seed storage proteins that were in this malt to produce or to result in amino acids and peptides in the hot water solution. So this then proceeds, and then after about an hour or so, that's stopped, and the residual seeds that's left over from the malt is filtered away, and so you've only got the soluble remaining wort, as it's called, which has got a whole lot of maltose, a little bit of residual dextrins and starch, some intact proteins, and proteins that have been digested down to amino acids. So this is a fantastic growth media for microbes, but it's prone to contamination at this stage, and that's particularly considering the ancient development of this biotechnology, right? When people have been making beer for tens of thousands of years, or 10,000 years at least, and they don't really want this to get contaminated. And so one way to avoid that is to then boil this wort, which sterilizes it. And then also often at this step, bittering agents like hops can be added. So that is added both for a sensory purpose, right? The bitterness of those hops can counteract the sweetness of the sugary wort, even in the final beer. But there's also some antibacterial, antimicrobial activity in hops or other things which used to get added to beer that can help preserve the beer as well. So that boiling then extracts out the hop acids and flavor compounds, and there's different ways that can be done to get different sorts of styles of beer. But then finally, that hopped wort is filtered and cooled, and then is perfect here, perfect media for inoculation of the yeast, which fermentation can then proceed. Right? So yeast then will use the sugars and amino acids and other nutrients that come from this wort, grow, produce more yeast, and as byproducts of that, produce ethanol, carbon dioxide, and also a wide range of flavor compounds that come from the process of yeast growth and fermentation. So then that fermentation, after a couple of days or so, depending on the precise style and conditions, is finished bright beer. And then typically that's filtered to remove the yeast and any residual solids, particulates. And then you've got bright beer, which can be carbonated and stored and sold. So I forgot my steps there. Yep. So there we go. We've got that whole process then through to the production of beer, right? So this is kind of an interesting bioprocess, and we've looked at this using systems biology approaches at all of these different angles here, right? So to understand the complex biochemistry and protein biochemistry and small molecule biochemistry that's happening during each of those stages. And that's been really quite fascinating to see that we can apply those modern tools in systems biology to really understand the details of this ancient bioprocess. But for the purpose of this talk here then, we've got this final beer, which is really quite molecularly complex. Because it's made from all of those different source materials, right? So it's made from barley and other grains, which lead to the presence of proteins, maltose, perhaps some residual glucose, some oligosaccharides that are still in the beer. Then we've got from the hops some terpenoids and other sorts of molecules. Of course, there's a whole lot of water and ethanol and carbon dioxide, but then there's also some secondary metabolites from the yeast. And yeast as well also secrete proteins and glycoproteins into this beer, right? So the final beer is a really particularly complex mixture of different sorts of organic and biomolecular molecules. So bringing this then around to yeast a little bit more, right? So modern beer production and consumption is still really dominated by the lagers in terms of the amount of beer which is made and sold and consumed around the world. Lager beer is far and away the dominant sort of beer produced. And there's of course some really interesting fungal biology that underlies the possibility of fermenting beer at low temperatures, which is what lager yeast can do. And in doing so, lagers have a really quite clean, crisp, perhaps boring palates, right? They're easy drinking, not particularly offensive, but neither not particularly interesting styles of beer. On the other hand, the other key type of beer based on yeast, in any case, are the ales. And here, the flavors which come from that yeast fermentation are really central to the flavor profile, the sensory profile of these beers, these ales. And so these are definitely much more interesting, and some might say offensive, but there's a whole lot of diversity there in the types of flavor profiles that you can get when you let the yeast secondary metabolism shine in there. And this is perhaps highlighted by Belgian beers, for instance, here, where there's a whole lot of really interesting yeasty farmyard sour type flavors that are in those beers. Now, over the last decade or so around the world, there's of course been a surge in popularity of craft beer and the diversity of craft beer producers. But and a key element to that growth in production and demand of craft beer has been the need to make something that's a little different from the generic lagers that dominated most of the world's beer production and consumption. And an easy way for craft brewers to do that was basically to make a standard simple ale and to add lots of hops. And this is great for craft beers to do because craft brewers to do because they don't need to modify their process particularly much. They can just make a standard ale, and they can add lots of different hops from different sorts of commercial providers. So in terms of a easy process to make in your startup craft brewery, just being able to make a standard pale ale or India pale ale with slightly higher alcohol content and then mix and match different types of hops is really quite easy. Easy to do, but it's also easy to generate some diversity in the flavors that come from that. So that's great, but it is relatively one-dimensional perhaps, right? And just relies on the hop flavor that can be added. And instead, of course, there's the potential to access this diversity of flavors that could come from yeast. And of course, that has been done, right? There are lots of different yeasts that can be used in beer production styles, and they can be highly diverse and give rise to lots of interesting beer flavors. For instance, these sorts of styles here, right, where they're found made all over the world, but particularly focusing in on this lambic style, which goes to quite an extreme length to access that environmental microbial diversity. And the way that it generally does that is beer brewers make some worts, right? So that extract of the malts with hops in there, a little bit of hops, and then they leave that open to the environment for contamination or inoculation by a variety, a mixture of different environmental microbes. And this can be done in various different circumstances. So literally out in the open outside, or perhaps in a more controlled enclosed barn, but nonetheless still quite open to inoculation from whatever is there. And then this sort of style of ferment needs to continue for quite a long time, because this is inoculated not only with yeasts, but also with a wide variety of other microorganisms, including diverse bacteria. And so the early stages of this fermentation can be really quite nasty and interesting. And it's only right at the end of fermentation, when the yeast becomes dominates that the product becomes palatable and safe for consumption. And this is also pretty, so this process can result in really interesting flavors, but there's a whole lot of lack of robustness here because of this environmental flexibility. And so instead, the idea that we followed up on was to use microbiological techniques to have clonal yeast to inoculate, still imparting some of that interesting microbial diversity, but with a much more controlled process, rather than having to rely on luck, basically, to have a nice inoculum. But of course, this is a little bit complicated, because most environmental yeasts really like eating sucrose, right? And in the beer making process, maltose is the key sugar which is present. And this maltose is a disaccharide of glucose that comes from the digestion of starch during that mashing and malting process. So this was one of the key elements that we suspected we might have some difficulties with in selecting for environmental yeasts that could produce beer with nice flavors, but that also were able to consume a sugar which perhaps wasn't their favorite natural carbon source. Okay, so then when Ed had started his PhD project, he basically jumped out into the field and went collecting samples of plant material, primarily from around the University of Queensland campus here in St Lucia in Brisbane. So it's a suburban campus just on the outskirts of the central business district, but we're lucky that it's very park-like as well, and so there were lots of diverse flora, that he could capture and collect, isolate yeasts from. So basically, he collected various plant material, took that back into the laboratory, and then enriched for fungi, for yeasts, right? So microscopic microbial fungi, and then used microbiology to ensure that we had single clonal isolates of yeast that we could then grow up again in that laboratory environment. So that then finally we had single pure culture isolates that representative of what was present in the semi-wild St Lucia campus. So then these yeasts we could identify using ITS sequencing to get a reasonable idea of the genus level identification of those. And we found that they were really quite diverse, right? So here in green, bold green, are the isolates that we could identify, just positioned within the fungi here. So there were quite some representation representatives from, for instance, from some genera, so the Pterospora, for instance, but they were by no means isolated to that, and they were really quite widely spread across the fungi. We did not manage to isolate any Saccharomyces, even though we're in a sort of semi-urban environment here. All of the, nonetheless, the yeasts were really quite diverse. And these yeasts that we did isolate, they've been previously described as being associated with plants and also with wild fermented beverages, so of beer and also of wine. So that was promising. And then we performed some initial screening to test whether these yeasts that we could have isolated actually had potential utility in fermenting beer, so growing on maltose and producing ethanol. And shown here are the results of those assays. So this is showing in both in terms of weight loss of the ferment. So that's the consumption of glucose to carbon dioxide. We can measure that conveniently with weight loss. And then we also use GCMS to measure the amount of ethanol produced. And this is on growth on wort, so with maltose as the key carbon source. And here we could see that there was a wide variety of abilities of these wild isolates to grow. So some grew really quite well and produced quite a lot of ethanol, whereas others, although they could grow well on glucose, they couldn't really grow at all on maltose. So there was a really quite wide variety of phenotypes in that space. And quite interestingly, even within nominally the same genus, right? So for instance, the Pterospora, all of these isolates here with arrows are Pterospora, but some grow very, very well and others very, very poorly. Even actually within the same nominal species, at least as defined by ITS, there was a really large diversity of phenotype within that space. So we could identify species, but we couldn't necessarily predict their behavior just based on that identity. We actually had to do these screening tests to isolate some yeasts that could potentially grow quite well on maltose and produce some ethanol. But we found some of those. And then the next thing that we proceeded with was just to confirm that those wild yeast isolates were actually able to grow on maltose as a key carbon source. We did this just by some metabolomic measurements here of the initial, so pre-inoculated worts. Could measure that, yes, it had quite a lot of glucose, a lot of maltose, and a little bit of maltotriose. After fermentation with a standard brewing yeast, this is used in most craft breweries, USO5, that could completely use the glucose and really quite effectively use the maltose, but not use the larger sugars like maltotriose. And then when we looked at these 3 Pterospora wild yeast isolates, we saw that all of those could efficiently use glucose. None of them could digest maltotriose. And 2 of them that were the ones which could effectively grow on wort, they could also effectively use maltose. Whereas this one isolate that could not effectively grow on wort, it couldn't grow because it couldn't use maltose effectively. So that was consistent with our growth assays, as confirmed also by that growth on specific carbon source, defined carbon source media. Okay, so we knew that these yeasts could grow on maltose as a carbon source. Next, we wanted to profile the small metabolites which they might be producing during that fermentation using untargeted LC-MS metabolomics. So we developed and used this untargeted LC-MS metabolomics pipeline and used it to basically identify the dominant secondary metabolites that were present in that spent media, or analogous to beer, after wild yeast fermentation. So when we've done that, this is the data here. So the abundance of particular metabolites shown here as a heat map. We've got a wide variety of metabolites, most of which change compared with after fermentation. So most of these sorts of compounds here, which are abundant in wort and which are absent after fermentation, are things like amino acids and sugars, which are nutrients that are consumed by the yeast during fermentation. And then we also have these molecules up the top here, which are not present in the wort, but are really abundant after fermentation. And those are the products of yeast growth, right? So some of these are things that the yeast are producing the secondary metabolites. And many of those are shared between the standard brewing yeast, USO5, and the wild yeast, the toral spores, but others are really quite distinct. And some of those that were of particular interest then are these compounds here, so tryptophan and ferulic acid, which were made in really quite high amounts by these wild yeasts. And these are compounds that have been associated with ales before. So in particular, these wild ferment Belgian ales or Hefeweizen, wheat beer yeasts, ferments which are more sort of farmyard, closer to wild yeast than standard lager or simple ale ferments. So this was great. It showed us that potentially these wild yeasts could produce interesting beers, but in a way that they could still produce them because they could grow on maltose as a carbon source. So this was fantastic, and we then, with some of those select isolates, proceeded to try and upscale growth and production of these yeasts. So here then we're showing the ability of these yeasts to grow. So measured here on the y-axis is the specific gravity of the wort before fermentation and then after fermentation. So this is basically measuring the sugar content of that wort. So initially there's lots of sugar in the wort, and then after the yeast have grown, they consume that sugar. It gets turned into ethanol and carbon dioxide, and so the specific gravity drops. So when we took this selected wild yeast isolate and grew it at small scale, so laboratory scale of some tens of mils or up to 5 litres or 100 litres, we saw that those yeasts were able to efficiently consume the sugars, so mainly the maltose, and grow. This was great. But then repeatedly, when we attempted to upscale these fermentations to 1,000 litres, the yeast just didn't grow effectively. They couldn't grow at large scale. And this was a bit of a problem because if we wanted to proceed to commercial production, we needed to increase scale. And so this was going to be a bit tricky. So we weren't entirely sure of the cause of this increase in or decrease in growth at scale, but we suspected that it was due to the presence of multiple environmental stresses on the yeast. We knew that they probably didn't like maltose as a carbon source, but they could grow on maltose when at small scale. And the other key difference here is pressure, or hydrostatic pressure. So when you go from a really small scale to a really large scale, that's one aspect of the environment of the yeast that changes quite substantially, is the amount of pressure, so the head of water on top of those yeasts, and that can affect cell biology in lots of different ways. So we tested and confirmed that individually the presence of glucose pressure or maltose pressure had no effect on the yeast, so they could cope with these stresses, but only if they were applied one at a time. Whereas if we applied pressure and forced the yeast to grow on maltose, then they had a substantial growth defects at small scale in the laboratory. So really suggesting that this effect here that we were seeing at scale was due to this combined pressure or combined stresses of pressure and carbon source pressure.
Ben, can I ask you a question at this point?
Absolutely.
Have you tasted?
Sorry, what was that again?
Have you tasted the beer at this stage?
Not at So the final beer, absolutely. But also at this stage, yeah, there were some so we didn't ever do sort of quantitative sensory analysis. But yes,
all right.
The head brewers at Newstead preferred to rely on their judgment of the quality, but they quite liked it at this stage. Yes. Yeah.
Oh, okay. So you do know that it is a good beer.
Yes, that's right. Yes, sorry, I didn't quite emphasize that here. Yeah, so the ferments here, yeah, so they actually smelled really quite delicious, right? So they sort of were really quite fruity and passion fruity. That was sort of the dominant notes, but yeah, also with the sort of hints of clove and peach. And yes, thank you for emphasizing that. The really nice thing about these ferments were that even with really low levels of hop, so I think a little bit of hops were added here just for sort of a little bit of background bitterness, but the main flavor that came through was from the yeast-driven secondary metabolites, and they were really quite deliciously fragrant and fruity.
Oh, okay, cool.
Yeah.
Thank you.
So yes, we really did want to make a reasonable amount of the beers. Yeah. Yeah. Right. Okay. Yes. So though unfortunately, although we wanted to make more of these, we were kind of stymied here because we needed to grow on, or wanted to grow on maltose, and the high pressure of large scale was making that difficult. And so to test whether it was indeed this dual stress of maltose and pressure which was causing this growth defect, we first experimentally at small scale removed one of those stresses. We did that by adding glucoamylase enzyme. So this is enzyme that will digest maltose back into glucose. And just to reiterate here, so when yeast will grow fine if there's no stress, or if there's just a single pressure or maltose stress applied, but with the presence of both of those, there's this growth defect. And then if we are to add glucoamylase in, that relieves that pressure, basically because we're removing the maltose and turning it into glucose. So we knew that if we added glucoamylase, we could basically remove this carbon source stress. And that indeed allowed us then to grow really efficiently, really efficient ferments at moderate scale. And so then that did let us produce a couple of small batches of commercial beer using this Torospora yeast, which was delightful and nice little finishing note to Ed's PhD, and that went really well. Along with this delightful decal here showing the jacaranda trees that there are lots of at UQ, and from which this Torospora was isolated. So that was fun and a nice ending to that commercial side. But we were really then interested in what was going on with this dual stress. And we found it quite fascinating that the yeast could apparently cope with either carbon source stress or pressure stress independently, but not in combination. And that suggested that there were some really interesting aspects of the cell biology of the yeast that were underlying that we really wanted to get a handle on. So to do that, we used our skills in proteomics to try and assess how the wild yeast were responding to these different pressures. So we could sequence these yeasts, get quite nice whole genome sequences of them to then be able to perform proteomic analysis. So we grew yeast at laboratory scale in glucose or in maltose as the carbon source, and then just at atmospheric pressure or with additional pressure applied, and then performed whole-cell LCMS proteomic analysis of the yeast under those different conditions. So when we compare the proteomes, quantitatively compare the proteomes of the same yeast grown under those different conditions by a principal component analysis, we see that there is really clear separation here depending on whether the yeast are grown on glucose or on maltose. So that's the key separation here. And there's, at least on this initial PCA, there's not so much separation based on pressure or no pressure. So glucose to maltose, that carbon source difference is the key defining factor in controlling the proteomes of these yeasts. And the key things that are changing in the proteome there are relatively well-established systems for utilization of maltose. So there's transcription factor networks and secreted and intracellular maltase enzymes that can take oligomers of glucose and that are alpha-1,4 linked and digest them down to glucose. So those are the enzymes that are present in or encoded in the genome of this particular Torospora isolate, and they are upregulated under growth on maltose, and that controls this difference here. So that was good, but we kind of understood what was going to be happening there with the carbon stress. And so we were more interested in trying to look at the more subtle differences that would more subtle changes to the proteome when we applied pressure to these yeast. And there were, although the changes were fewer and less in abundance than the changes on carbon source, there were quite a number of proteins that were changed in abundance when we added pressure to these yeasts. And we could perform GO term, gene ontology term enrichment on these proteins And there was a list of those Go terms then that were enriched in these proteins that were less abundant without the addition of pressure. Now, most of these are proteins that are associated with the ribosome, and that basically, in terms of yeast biology, that basically correlates with growth rate. So the faster yeast are growing, the higher proportion of their cellular proteome is dedicated to the ribosome, because they're little balls of ribosomes that just pump out more proteins to make yeast. And so here, if we're seeing reduced ribosome proteins, that's basically just saying that the yeast are growing more slowly under this pressure stress. So that makes sense. It's not particularly interesting. But the one term which you did leave out that was separate to that was the esosome. Now, I didn't really know what esosomes were, but they are kind of analogous to caveoli in high eukaryote biology, but they are plasma membrane domains that are somewhat enriched in sterols and also enriched in particular subsets of the proteome. And we found 3 proteins in particular that were associated with this esosome Go term. The 2 sort of canonically esosome-associated proteins, PIL1 and LSP1, they're homologs. And then also PMA1, which is the major plasma membrane ATPase in yeasts that was also associated with this Go term. And all of these proteins were reduced in abundance under pressure conditions. So esosomes have been studied a little bit over the last years or so. And one of the key roles that they have, so the esosome itself is a little plasma membrane invagination that's similarly enriched in particular classes of lipids. So for instance, the sphingolipids and sterols are enriched here. And PIL1 and LSP1 are the key structural proteins that form these membrane curvatures and invaginations in the membrane. And then they're also associated with particular classes of plasma membrane, integral membrane proteins, including these APCs, which are key amino acid transporters. So the esosomes can act as stores of these amino acid transporters where they're not active, but they are still present at the plasma membrane. And then under conditions where the cell needs to uptake amino acids, those amino acid transporters can then rapidly transition into the neighboring plasma membrane where they're active and have proton-coupled amino acid imports into the cell. And so then it would make sense also that PMA1 is associated with this process, because PMA1 is the major ATPase that generates that proton gradient, so extracellular protons that can then be coupled with nutrient transport, nutrient import, and in this case with amino acid import. So this was what was sort of established in the literature about esosomes. And in our data, we saw that PMA1 and these key structural components of the esosome, PIL1 and LPS1, were less abundant in conditions of pressure. And so we thought that perhaps there was some link here with amino acid nutrient uptake or utilization by yeast under these conditions. And so we wanted to investigate that further. And so we went to metabolomics, so LCMS measurement of the concentration of particular amino acids inside the cell. So we're measuring intracellular amino acid concentrations here. And first we measured here just in Saccharomyces cerevisiae as a model, the amino acid concentrations in standard growth conditions, and then applied extra pressure to that standard Saccharomyces cerevisiae strain. And we saw that there was a subtle but significant reduction in intracellular concentration of a subset of amino acids. So particularly isoleucine was significantly less abundant inside these yeast cells if we apply pressure to the cells. And then if we look in combination with PIL1, here we couldn't. So we had seen rather in our experimental proteomics that when wild yeast was grown under pressure, we saw a loss, a reduction in the abundance of PIL1. So to try to measure that in Saccharomyces experimentally, instead we measured a delta PIL1 in Saccharomyces. when we measured the amino acid concentrations there, we saw that indeed there was an increase in the concentration intracellularly of these amino acids. So it's consistent with our wild yeast experimental data, suggesting that if you reduce the concentration of pill 1, either with physiological regulation or by knocking out the gene, that will serve to increase amino acid uptake and hence the intracellular amino acid concentration within the yeast. And if we combine this delta pill 1 with pressure, we see that pressure is again reducing the intracellular amino acid concentration, but it's back to a level now which is equivalent to the concentrations of wild type yeast without any pressure. So that is, pressure is reducing the ability of yeast to uptake amino acids, but pill 1, if the abundance of pill 1 is reduced, then that lets the yeast increase their amino acid uptake and can compensate for that extra pressure. So we think then what's happening, our model of what's happening, tying all of that together in the wild yeasts, is that there's an interaction here between amino acid uptake and carbon source utilization that's underlying this dual stress that we see in the wild yeast. So under normal conditions here, PMA1 is able to acidify the extracellular environment, and these amino acid transporters can then transport amino acids from the media intracellularly. But that under conditions of high pressure, we don't exactly know why, but that we can speculate that the environment within the lipid is changing, lipid bilayer is changing here, or that these proteins just aren't working as well. But under conditions of high pressure, these amino acid transporters are less efficient. But that's okay, because there were some of those amino acid transporters that were hanging out in the esosome, and they can be released under these regulated conditions to compensate for the relatively inefficient action of those amino acids and still allow a high concentration of amino acids inside the cell. But then if at the same time we have carbon source stress such that PMA1 isn't provided with sufficient intracellular glucose, then this acidification doesn't work so well, and then we have this synergistic impact here, which then leads to lower concentrations of amino acids inside the cell. So we haven't tested all elements of that model completely, but independently, that all comes together to perhaps provide us with a model of what's happening here that explains why these wild yeast are able to cope with those environmental stresses individually, but not synergistically. And that's then directly relevant to using these and other wild yeasts in industrial settings, where not only the underlying biochemistry is important, but also the ability to grow these yeasts at industrial scale is really quite important. And so I think there's interesting avenues for pursuing and understanding this nutrient availability and metabolic control aspect of yeast biology beyond Saccharomyces to enable industrial use of a variety of these different wild yeasts. Okay, and that brings me to the conclusions. Again, I should really thank here and acknowledge the work of Dr. Ed Kerr, who did all of this experimental work, and highlight again here that, yeah, we have this shown that the simple workflow works really nicely for isolating, identifying, and assessing the fermentation capability of wild yeasts, that it's possible to find some wild yeast that can make unique and interesting beer, although I should highlight here that it's only sort of a 1% or so of the wild isolates that we found that were made it through our pipeline and screening, right? So this explains why it's really quite difficult to just do a wild ferment, at least in subtropical Brisbane, right? You really need to be quite selective about the yeasts that are used in fermentation. And then that we could use systems biology to address and understand the metabolic challenges of upscaling fermentation. And more excitingly, I think that we've only looked here at a really small aspect of the metabolic diversity of wild yeasts, and there's a whole lot more that remains to be discovered and used. Thank you all very much.
Thank you, Ben. That was absolutely fascinating. Do we have questions from the audience for Ben? Or I raise a bunch. Naral.
Thanks so much, Ben. That was really, really interesting. And I don't even drink, and I really loved it. So it was really good. I've got a question about, so you've got some metabolites, like your tryptophan and your ferulic acid. Are they associated with particular yeast types, like yeast species, or do you find that there is some redundancy, like if you've got a mix of yeast and some are more dominant or others, like in the microbiome, how sometimes you knock out particular species, but they're a bit redundant, something else will come up and make those same metabolites? Do you see any of that kind of profiling happening?
Yeah, so in pretty much everything that we've done so far, including all of this data and including the metabolomic profiling, we've only considered pure cultures of yeasts, right? So we've taken yeast from an initial mixed culture in the environment, but potentially from different environmental locations, isolated them so that they're clonal, and then grown them up. But when we do that, we find that there's essentially a mixture of conservation of the production of those different metabolites. So there are some metabolites that are essentially universally produced, or on the flip side, there are some nutrients that are universally consumed. Right? So glucose, for instance, key amino acids are essentially universally consumed. And then in terms of the metabolites that are produced, so for instance, things like those ones, the tryptophan and ferulic acid, they're actually quite commonly produced by yeasts across different genuses. But with some quantitative differences. Off the top of my head, I can't think of others or quantitatively exactly how much diversity there is there. But in summary, there are definitely some of those compounds that are produced by most of the wild yeast we've isolated, but definitely not all. And then on the other hand, there are also some yeast that at least quantitatively, have really quite distinct profiles.
Yeah. So, awesome. Thank you.
But it's also a really interesting question that you raise about the ecology of the system, and that's something that we're certainly really interested in looking at in the future, is about how these yeasts and other microbes, or how these yeasts behave, not just in isolation, but in co-culture with other yeasts and potentially also with other microbes.
Thanks, Ben.
Yeah, actually, Ben, I was going to ask about that. Is there any value in getting a yeast in there that would digest the maltose for the next yeast to use, rather than putting an enzyme?
So conceivably, exactly. I mean, and there's different solutions to that, I guess, depending on what the question is. Right? So, in an industrial type environment, it's probably more efficient to either have a feedstock which the microbe will grow well on, right? So to not be so fixated on using worts with maltose, right? But instead, just if the purpose was to grow this particular yeast, you can select the media such that the yeast is going to grow well, right? So to have like a glucose-rich carbon source, either from glucose to start with, or by adding an enzyme which is really cheap and well controlled, rather than add an additional microbe to the mix. But that ecological interactions between species, depending on what other constraints there might be, or to investigate actual real environmental ecological interactions, that's also really interesting. Yeah, absolutely. And there are certainly some yeasts that at least have some capacity for secreting glucoamylases that can degrade oligosaccharides or even starch into maltose, into glucose. Sorry. So there's some capacity for that. But yeah.
Yeah, because I guess the feedstock has to be barley, doesn't it, to make it beer, or can you use different kinds of feedstock?
Yeah, that's right. Yeah, I think it depends where you are in the world. It's more of a branding and legal definition, actually. So I think in Australia, it's relatively flexible. But for instance, in some parts of Germany, you must have beer or wheat, and you must not have anything else in there. Otherwise, you can't call it beer. It's still able to be made and sold, as long as it passes the food quality things, but it can't actually be called beer. So in Australia, there's less regimen around what a product has to contain for it to be called beer. and some might argue that the standard generic lagers that are really popular maybe don't even meet that characterization, right? So there's a lot of flexibility there. But exactly, that's what we were after here, right? And especially working with Newstead, they really wanted to make a product which was which didn't take any shortcuts and was a real sort of craft beer product. So that's why we used those ends.
Are there other questions from the audience? If not, I might ask another one if I may. It seems like it's a really random process in Belgium to be using these wild ferments. Do you think they have to throw away a lot of stuff? Because like I can't imagine. Or do you think that actually they're not that wild? That in fact.
Yeah, I think it's probably a combination of those, exactly. So yeah, I don't actually know. I haven't seen any reports on like the proportion of ferments that have to be discarded because they just fail or get contaminated or never proceed. I'm not sure about that. That's a bit of a black box there. But yes, I think it's also the case that they're open for inoculation, but typically in a quite controlled environment. So it's inside barn-like spaces that have been used for brewing for a long time. And so not animals are flying past and dropping stuff in. Instead, it's a relatively well-controlled environment, and probably that environment has been inoculated over the last centuries with microbes that have been selected to grow well in the wort, right? And so probably it's selecting for mixed cultures which will produce a good quality product in the end relatively efficiently, right? But just sort of so like almost there towards microbiological clonality, but just instead within a relatively controlled environmental setting.
Yeah.
But there are people around the world, people who I've heard of who literally do take their wort and leave it outside overnight, right? So
just hope for the best.
Yeah, that's right.
That's like a very risky thing when you think of what else could.
Exactly, that's right. And I think the key thing there that's been reported anyway is that it just takes time. So initially there's the bacteria take over the culture, and the ferments after weeks or months or so is definitely disgusting and probably not very good to drink or not very good for you either. But then the yeasts take over the ferments and convert a lot of the small volatile fatty acids into ethanol, and then the preceding ferment then, when it's controlled by those yeasts, becomes high in ethanol that is toxic for the other microbes, and so biologically controls their abundance. And so the final ferment then, literally after years, is well controlled and safe, but still with hints of the flavor profiles that come from the diversity of microbes that were there to start with.
Right, but it actually takes years, does it? It's really long.
Yes, yeah, that's right. Yeah, so 2 years or so, I think, is relatively standard for those ferments. Yeah.
It's a long time to wait for a beer.
Exactly, that's right, especially in the context of a small craft brewery, right, where you don't really want to be storing things for a long time before selling them.
That's right. It might just be horrible anyway.
That's right, exactly.
And just one final question. Were you surprised you didn't get any Saccharomyces? Is that normal, do you think?
Yeah,
like that?
I mean, we were a little surprised, but I think it's relatively consistent. So the studies which have been done on the microbiome during wine ferments, for instance, kind of shows similar sorts of profiles that and that there are Saccharomyces that are present on grapes, but they're quite low in abundance. And there are many other species that are more dominant on the actual fruit. But during the course of the fermentation, it's the Saccharomyces that really become dominant and then control the fermentation in the latter part of the ferment. So again, it's consistent with what we see, that Saccharomyces are quite low, they're quite rare environmental fungi, but they're particularly well suited to coming to dominate industrial fermentations.
Right.
Yeah. And then that's probably why they've become such a common industrial microbe.
Yes. What was it? what was it, Taurus?
Tyrrellospora. Yeah.
Yeah. Is
They're-
that used anywhere else?
So it's rather common in grape associateds. So it's a commonly grape associated microbe, and it's present as part of the co-culture in almost all wine ferments because of that. And it has also been used. There's a couple of other applications of isolation and use of Tyrrellospora isolates for like kind of similar sorts of production and fermentations. So specifically slightly lower ethanol ferments. Because back at our initial screening, we found that most of these wild isolates produced less ethanol, or they produced ethanol less efficiently than Saccharomyces did. And that's a nice outcome, right? If you have a fermentation fermentation product, a yeast that can fully ferment, produce interesting flavors, but with less ethanol than Saccharomyces, then that's a nice market niche.
That's true. Yeah, that's becoming a big thing, isn't it? The alcohol beer.
Yeah.
Well, thank you so much. If there's no other questions, I guess we'll just all thank Ben again.
Thanks for joining us today. Please subscribe for more great content, and we look forward to seeing you again next time.